My name is Robert Boyle. I'm a clinical reader here, and I'm going to be speaking with you this evening together with Professor Adnan Custovic and Dr. Marta Vazquez-Ortiz. We've got a really exciting lineup this evening. I'm going to start with a few points about studying allergy at Imperial, the postgraduate certificate, diploma, and master's courses we have here. And then Adnan and Marta are going to give some cutting-edge updates on two really interesting areas of allergy research, which are particularly strong at Imperial. Imperial is a top 10 university worldwide, currently ranked 6th. It varies from year to year, but we're in the top cluster of universities globally in most global rankings. Our particular strength is research, and immunology research is a big part of Imperial's offer. Allergy really has a very special place in Imperial. Many people refer to Imperial as the home of allergy, and that's because allergen immunotherapy, one of the main treatments for allergy, was discovered here. The first randomized controlled trial of allergen immunotherapy was conducted by the late Dr. Bill Franklin in the 1950s. And we've had a stellar cast of allergy researchers here, most recently the late Professor Barry Kay. And we really have a very strong faculty here, and that's reflected in all sorts of rankings. If you look at the World University rankings, when they do it by when they rank universities by topic, then in allergy we come up at close to top in the world. We have a really strong faculty, and that really is the core of our teaching offer. We have quality here, really top quality names, people that if you've been involved in allergy, you'll recognize many of the names, the top contributors in many specific areas. The syllabus is really broad. Where we do have less strong areas, we bring in international speakers to the master's courses. So it's not just the Imperial faculty delivering it, but the Imperial faculty are the core and account for the quality of the offer that we have. It's a very flexible course. We have people from all over the world joining. We have an online offer and a blended offer with some time on campus and some online. And we're very student-centered. We listen to our students. It's a very active learning environment. Students do a lot during the course. It's quite intense. It's quite heavy, but you learn a lot and you become very expert during the course. The sorts of areas where Imperial is currently very active in research is, of course, still rhinitis immunotherapy, which is, as I said, was discovered here over 100 years ago. We have a very strong offer in asthma with some of the world's most highly cited asthma researchers at Imperial. Allergy epidemiology also been very strong for many decades. We have increasing strength in eczema or atopic dermatitis and primary prevention of allergic diseases. And what you're going to hear a lot about today with Professor Custovic is learning from cohort studies and aggregations of multiple cohort studies, the application of bioinformatics to those, and how that helps us to tease apart different disease phenotypes and get better ways of diagnosing and predicting outcomes in those. Where do our students come from? Most of the students are doctors, but by no means all of them. We always have some dietitians, nurses, biomedical scientists, people from industry sometimes, epidemiologists, so people from different areas of clinical science. The doctors come from various disciplines, and the same for the nurses and the dietitians and the biomedical scientists, but often with some sort of allergy, immunology background. So doctors who are already working in allergy or working in pediatrics and want to develop an interest in allergy, or in primary care, dermatology, respirology, ENT, so various areas. We've had people from really all walks of clinical and non-clinical science, but the majority would be from a medical, nursing, or allied health background. Over the last 100 students, about half of them have come from the UK, about half from other countries. We often have students from Ireland and the European Union, but quite a lot of students are from other countries around the world. And particularly in the last few years, since we've had an online-only offer, it's been much easier for people to join the course from all sorts of time zones and parts of the world where it might have otherwise been quite difficult to travel to London and take part. What do the allergy MST students do? Well, we've been offering this course for about 15 years now, and it's really nice to see some people progress into academic careers. So quite a number go on to do MDs or PhDs, and some of our early cohorts, some of the students are now professors at different faculties around the world. But there are other ways to progress with an MSc. MSc can be useful just within a clinical role, allows you to move to more of a leadership position within a clinical role, to develop a more concrete area of interest and allergy, or a private practice. It definitely can give other job opportunities within industry, or research, clinical research or non-clinical research as a medical advisor, etc. So there's quite a range of things that people find the Allergy MSc has given them a springboard to in their career. What does the course look like? So the first year, which is if you do the first year on its own, that's called the postgraduate certificate in allergy. That's a qualification in itself, and it's largely taught course in 3 modules. The first is a scientific basis of allergy, which touches on it just helps students understand what allergy is, what are the immune mechanisms understanding allergy, and how do they relate to the clinical manifestations and the treatments that we use. So it helps you understand the clinical practice of allergy, why things are the way they are. The second term is about the practicalities of the diagnosis and treatment of allergy. Allergy diagnosis, not always that easy. We help you understand that, and we look at the different areas of allergic disease, so skin disease, drug allergy, respiratory disease, the basics, what goes into diagnosis and treatment of those areas. So between those 2 modules, you will have got a really good grasp of why we do what we do in allergy and what people do, what's current best practice. The third module is more thinking outside the box. It's going beyond that. What's coming over the horizon? What are the new things that we're going to be practicing in allergy in the next 10 years? What are the maybes? What are the big research endeavors at the moment? And what are the things that people are struggling to understand how to put them into practice at the moment? And all 3 of these modules have, as I said, a lot of active learning, a lot of pro-con debates, presentations, group work, lots of active learning, discussion, debate with the excellent faculty that I touched on at the beginning. The third module is also, we get you to start to think about research, try to understand research, try and think about designing your own research project. And that's particularly helpful for those who are going to spring on to do a full allergy MSc program and complete a research project. So it's about cutting edge and also developing some research skills. The second year goes into more depth, and it's 5 different modules which all go into great depth with world-class speakers and learning sessions. The first is about food hypersensitivity. That's both immediate delayed type food hypersensitivity and all its manifestations. Second and third are about allergic respiratory disease. That's again rhinitis, immunotherapy, and asthma. And then the fourth and fifth are about allergic skin disease and allergies that affects young people. And so those build on the learning in the first year. And if you complete those first 2 years, you come up with a postgraduate diploma in allergy, which is a slightly higher level certification than a postgraduate certificate. And majority of people who join the course want to do the full master's course, and most people do get through that. And the master's component can be done in parallel in year 2, or it can be done as a separate year 3 activity. And this is a research project. And the research project is you have more than one supervisor. So you have a local supervisor wherever you're working, and then a co-supervisor from Imperial. But some people want to do their project at Imperial, and they have one of us as their primary supervisor, and then someone else as a co-supervisor, often a local person. So and the research project is, so the whole thing can take either 2 years or 3 years. What does it cost? The cost varies depending whether you're doing an online or a hybrid online and an in-person course. Whether you're what's called a home student, so that's UK or Irish based at the moment, or overseas, so that where your nationality is elsewhere. And it depends whether you're doing the postgraduate certificate, diploma, or MSc. So the course fee can range from around £7,000 for a home student doing the postgraduate certificate to over £40,000 if you're an international student doing the hybrid, in-person and online full MSc program. So quite a variation there. we're fortunate enough to have some bursaries and scholarships, so we're able to support students, and there's a selection process for that. So people who want to apply for financial support, we're able to support some students, and there's a deadline for application at the end of May. Usually, we're able to support around £7,000 for one person who's doing the full MSc, and then we have a number of smaller grants of 2,000 to 3,000 per person. We need to await confirmation each year that we're going to get that support, but we would be hopeful to be able to do that again this year for people who apply for some financial support. The core areas of research at Imperial are around mechanisms of immune tolerance and anaphylaxis, endotype discovery and diagnostics, primary prevention, environmental control, and allergen immunotherapy. And I'm not going to talk anymore. I'm going to hand over to Professor Adnan Custovic and Dr. Marta Vazquez-Ortiz, who are going to talk today primarily about endotype discovery and diagnostics and why they're important in allergy. Just to say, this isn't the last chance to ask questions about the course. We have an open day on Wednesday, the 5th of June. So put that in your diary if you're not sure, if you didn't get enough information about today. There's much more opportunity to post questions and have active answers rather than just listen to us highlight our research activities in that session in June. So I'm going to allow Professor Custovic to hand over now. If you've never heard him talk, this is a great opportunity, because if you ever hear Professor Custovic talk, you will never forget it. He's probably the best known and most memorable lecturer in allergy in the world at the moment, and we're absolutely thrilled that he's part of our faculty and has managed to find time to
don't overdo it. Don't overdo. Thanks very much for that.
Have I bigged you up enough, Adnan?
Huh?
Have I bigged you up enough?
Yeah, I hope you can see my screen. My
Lovely, yeah, thank
Teams
you.
is playing a little bit, but I mean, what I want to talk about is topic that has been very close to my heart for like 35 years, is the issue of whether we can understand progression from preschool wheezing to school-age asthma. I mean, you may ask the question, well, why on earth is this important? Well, this is important because our patients and families identify this as a key unmet need for parents of children with preschool wheezing. Hilary Pinnock, within the context of the new European Respiratory Society Task Force for Preschool Wheezing, has done fantastic sort of workshops with parents of kids with wheezing. There are only two questions that they're interested in. Question number one, does my small child have asthma? And to that question you have to answer, I don't know. And will my child still have this disease in 10 years' time? And you can say, well, unfortunately, I do not know. So what is the background here? I mean, the real problem with preschool wheezing, it's hugely heterogeneous. And it's heterogeneous both in its cause and in its cause. What does it mean? Well, there are different types of heterogeneities. One is the way we would describe it as circular heterogeneity. So that's heterogeneity in terms of time course, both time and onset and progression and its development. And the second one is etiological. Etiological means, well, sometimes very similar symptoms are underpinned by very different pathophysiological mechanisms. All of you who have dealt with small preschool children with wheezing will know that in some of them, in many of them actually, up to 30%, wheezing will remit by mid-school age, and we refer to them as transient wheezers. In others, their symptoms are going to persist, and we call them persistent wheezers. Some of these persistent wheezers may subsequently be diagnosed with asthma. But despite this clear heterogeneity of life course development, mechanisms, and late outcomes, when they present clinically for the first time, the clinical presentation at the onset of symptoms is identical amongst those whose wheezing is going to resolve and those whose wheezing is going to persist. And this is the reason why it's really difficult to predict the course of wheezing in these individual patients who we will see in clinic, and accurately define those who will stop as compared to those who will continue, thereby not being able to answer these two questions that their parents are troubled with. And on top of that, the mechanisms that are associated with either persistence or remission are really poorly understood. And Bob hinted at the areas that we're interested. We're interested in the area of modern data analysis, of how data analysis techniques applied to large data sets can help us understand human disease. And what you all know is that our ability to collect data has increased exponentially. We now can collect vast amounts of longitudinal clinical data, which is coupled by high throughput technologies, various omics technologies, and this gives us an opportunity, at least an opportunity, to understand the complexity of the developmental pathways of childhood wheezing disorders, which ultimately may or may not result in persistent disease. The problem with huge data is it's tricky to process it. It's even more difficult to analyze it. And it certainly is not easy to interpret it. I mean, sometimes, when you see the amount of data that you had, you kind of step back and you think, God, what is the question that I'm asking? And it is within this context of big data that I think modern data analysis techniques such as machine learning and artificial intelligence that you will have heard in the news every single day, may actually offer a part of solution. So what's machine learning? Well, machine learning is a tool. It's a fundamental technology that is required to meaningfully process the data that are far too big and far too complex for our brains to comprehend. When you have 10 patients with 3 variables, it is easy to see the patterns. When you have tens of thousands of individuals with millions of variables, any pattern is impossible to observe. And it is within this context that machine learning models can help, because they can digest, they can process these large amounts of data reasonably quickly and identify underlying patterns which our eyes cannot see and our brain cannot process easily. So it really is trying to understand the latent structure within these data sets. Now, there's a problem because people say, once we find these clusters, classes, you name them, however you want, we have found the endotypes of disease. These correspond to biological mechanisms. Well, you have to take this with a pinch of salt. The patterns within any data set may or may not correspond to underlying pathophysiological or biological mechanisms. You then may proceed and say, well, can we really trust the scientific discoveries based on machine learning? Well, you most certainly must verify them. And these are largely hypothesis-generating techniques where any findings you then take and then carefully check in meticulously designed experiments. When you think broadly about machine learning in childhood asthma, you have few different approaches. Generally in medicine, you have few ways of approaching how you use these complex techniques. One is predictive machine learning. So it's very simple. You make predictions. And you're really not interested in explanation of prediction for as long as you can very accurately predict what is going to happen in individual patients. So, for example, you see a 3-year-old and you say, well, I want a model that will tell me whether this child is going to have asthma in 10 years. And if I have a model that can predict it with 100% certainty, it's great. I don't need to know why. So this is pretty much, how some of the applications in machine learning, particularly in terms of image recognition, are currently working. And the approach is correct, and it's fine when you're building automated system where you are not particularly interested in explanation. Now, most of you who are here are curious. And for me, prediction per se, without actually understanding why and how any particular algorithm can predict outcome, is not fully satisfactory. And that's where exploratory machine learning comes into place, where goal is to explain or understand the observed patterns within the data and understand the processes that may give rise to the data. So the core idea behind model-based machine learning, which is what we have been engaged with for now 15 years and probably for the next 2 decades, is that you can make assumptions about the problem that you're studying in explicit form, in form of a model. And then you can follow it up and understand it. It's really great explanation of this approach in very recent textbook by John Wynne, one of our earliest collaborators from 15 years ago, about model-based machine learning. So how can we use this in asthma context? So I'll give you examples of both predictive and explanatory machine learning. So predictive one, yeah, simple. Can we predict in an individual child when they are 2 to 3 years old whether they will have asthma? And there's a really interesting question here. Question is, well, what the hell do we mean by whether the child is going to have asthma? And are we really trying to predict unpredictable? What do I mean by trying to predict unpredictable? Well, very recently, sort of within our consortium of UK birth cohort called Stella or Unicorn, where we have 5 birth cohorts, 16,000 individuals followed from before birth to now early adulthood, working in Southampton, Dilini Kotawala with Hassan Arshad and John Holloway, asked the questions, can we use machine learning applied to data, clinical data that we collected at age 3 to 5 years to predict whether the child will have asthma when they're 10. So in a series of models, and I will not bore you to death with what these models are, she started with about more than 50 candidate predictors, and then used machine learning to select the variables of particular importance. And indeed, 12 of them were very informative. For any one of you doing clinical practice, they will not come as a surprise. Preschool atopy, preschool cough, preschool wheeze, polysensitization, frequent nocturnal symptoms, socioeconomic status. Yeah, all of these were really important in predicting whether the child would have asthma. And indeed, this machine learning model was much better than asthma prediction indices or any other clinical predictors that have been published but actually never used in clinical practice. Why they were never used? Because they're simply not particularly useful. So how good was this model in terms of prediction? Well, you can put a brave face and say, hey, our machine learning approach is much better than existing regression-based model, with a good ability to rule in asthma and with area under the curve of 0.82, which quite frankly is not good enough for rolling it out in clinical practice. What does it mean, very good ability to rule in asthma? Well, it can tell you, roughly speaking, that 3-year-old child who had more than 3 episodes of wheezing, frequent exacerbation, who is sensitized to multiple allergens, is likely to have asthma. I would say you don't really need machine learning model. You need a little bit of common sense and clinical thinking, and you will come to the same conclusion. The real problem is, even amongst those children with these features, more than half ultimately do not have asthma. So we thought, okay, this is a good starting point. We can beat everything that is there, but cannot roll it out into clinical practice. So perhaps if we include further information like genetics, it will improve it. This is how good genetics is in predicting childhood asthma diagnosis. So all of the genetic variants, all 132 of them ever associated with asthma, have really poor predictive ability in terms of predicting the asthma diagnosis. So all genetic information, quite frankly, is pathetic. Now, if we add this genetic information and then epigenetic information to all clinical information, surely we will do better. But we actually not. Well, we have moved area under the curve from 0.82 to staggering 0.84 when we included the entirety of genetic and epigenetic information. So why? Why is it so poor? And the real problem is what we're trying to predict, asthma, is highly heterogeneous. So these are some of our very recent data, looking what hides under the umbrella doctor diagnosis of asthma. And 1/3 of these children have persistent wheezing, 1/4 of them have late onset wheezing, about 1/4 of them have intermittent wheezing, 6% of them have never ever wheezed. and 6% of them are transient wheezers. And you may say, well, wow, if all of these are actually underpinned by different mechanisms, it will be very, very, very difficult to predict them. So surely what we should be trying to predict is not the diagnosis of asthma, but those different mechanistic subtypes. And it is really interesting that, even amongst children with persistent wheezing who have wheezed year on year from birth to age 18, thirds do not have asthma diagnosis. Quite striking. There's an interesting research question there. If there's anybody who wants to do a master's project, this is a project waiting for you to do. So, okay, we cannot really predict asthma in preschool age with any sort of level of certainty. So can we understand the heterogeneity? And this is the story of wheeze phenotypes. And every clinician will know about wheeze phenotypes. This actually is a paper that really inspired me to go into this area. It's fantastic. It's Nicola Wilson, 35 years ago. 35 years ago, it's as fresh and as good as if it has been written yesterday. Look at this. Description of wheezing illness has always been confused. Tick, exactly the same as now. Justification and implication of classifying syndrome asthma in terms of clinical patterns and response to viral infection is something that we need. Tick, we're doing it now. The association between bronchiolitis and asthma remains contradiction. Yeah, it still is. And for the first time, she actually introduced the term virus-associated wheeze, which was absolutely great. And then our friend and collaborator, Fernando Martinez, tested this as a hypothesis in Tucson study and described predetermined patterns as transient wheezing, late onset wheezing, and persistent wheezing. The question is, okay, he hypothesized the existence of these and then managed to find patterns in the data. But what do data itself say about patterns of wheezing through childhood? Now, this is the first, what people call unsupervised, data-driven analysis, trying to see the pattern. The first, actually, analysis published from our stellar birth cohort consortium, which suggested that this is a little bit more complex than our hypothesis-driven analysis would suggest. And then to address this in sort of mother of all unbiased analysis of childhood wheezing, we mapped the progression of wheeze from birth to adolescence in all 16,000 participants in our birth cohorts from birth to adolescence, describing the structure with persistent wheezing, two resolving classes, late onset wheezing and never wheeze. Now, there's a word of caution here. People talk about transient wheezing as benign. The reality is each one of these wheezing phenotypes, including transient one, is associated with impaired lung function in adolescence. You do it with one data-driven model, you do it with another data-driven model. Every single wheeze phenotype is associated with impaired lung function. If there's one thing I want you to remember, it's this. There is no such thing as happy wheezer. There is no such thing as benign transient wheeze. Why? Because the level of lung function here at age 24 is the strongest predictor, not only of COPD 40 years later, but also of premature death of all causes, cardiovascular and metabolic morbidity. The question is, are these different phenotypes different diseases? And you may twist it upside down and say, hey, if they're underpinned by different genetics, surely they are. So this is very recent work. This is where I'm bringing you to what we're doing now and what we will be doing in the next few years. Asking the question, okay, are these wheeze phenotypes really different diseases? Now, for those of you who know about genetics, you will be familiar with Manhattan plots. This is something very different. This is one of the first examples of multinomial GWAS. We're not interested in genetics of asthma, but of different wheeze phenotypes. Each circle is different phenotype: persistent, transient 1, transient 2, late onset. And staggeringly, genetic associates of these different phenotypes are phenotype unique. There are 40 SNPs exclusively associated with persistent, no other phenotype, 25 with remitting, 32 with late onset wheezing. Genetic architecture is different. Is there any shared architecture? Well, here in red are signals, genetic signals specific for persistent wheezing, blue specific for transient, green are shared. Purple are shared in opposite direction, but there is no green or purple to be seen. There's just uniquely phenotype-specific association. When you drop your significance level to 10 to -5, you start seeing these green hints of shared genetics. And it will not come as a surprise to anybody who knows anything about genetics of asthma. This is 17q21. Okay, there were also two hits that were significantly associated at genome-wide level, so less than 10 to -8, uniquely with persistent wheeze. 17q21 previously described, and brand new hit in chromosome 9. Nobody has described it. What the hell is it? Where is it? What does it mean? Well, it is close to the gene called annexin-1. But being close to it doesn't necessarily mean it's the same. So we were really interested to see what is the causal SNP. And two of these SNPs that were associated were located downstream of annexin-1. So perhaps this is the gene of interest. So what we did then is a series of very carefully designed studies, first starting with promoter capture high-C interactions. This is work by John Curtin, where he has shown that as chromatin sort of folds, the SNP, one of these SNPs, associated with persistent wheeze, actually physically interacts with an annexin-1 promoter. Bang! Probably positive variant. Interestingly, the same variant that is associated with persistent wheezing is also resulting in diminished annexin production. Even more interesting, what the hell does annexin do? Well, it actually interacts with genes that are associated with asthma, like IL-4 and IL-13. So suddenly it becomes really interesting. What do we need? We need replication. But this is hypothesis generating. There aren't any wheeze phenotypes available around the world that are similar to these. So we had to move to mouse experiments, work by Claire Lloyd, really fantastic work trying to understand what would be the mechanism by which annexin-1 can be associated with persistent wheezing. So she used a mouse model of how dust mite induced allergic airway disease, take it with a pinch of salt, but it is very interesting that following dust mite challenge, there's marked increase in expression of annexin-1, both at mRNA and at protein level. Now, she then created knockout mice, and in a knockout mouse, this knockout mice have significantly worse lung function, allergen-induced lung function, a hell lot more eosinophils, T2 lymphocytes, more IL-4, more IL-5, more IL-13. So now it becomes really suggestive that this is a proper hit associated with persistence of wheezing and provides a new target for intervention, because the pro-resolving annexin-1 may play a role in regulating pulmonary responses to allergens. So what you can see very clearly here is the power of approach that we are trying to implement here, which is applying modern data analysis to understand patterns within large clinical data sets, follow this by high throughput omics technologies and careful experiments to prove causality. Finding the structure is not enough. Trying to explain it is what we're trying to do. So you may say that's absolutely great and these patterns are stable. Always take it with a pinch of salt. Why is the validation important? When we replicated our finding in South African birth cohort, the structure of wheezing illness is very similar to what we described in the UK. But unlike UK, where rhinovirus and allergens are driving persistence of wheezing, most of this is uniquely related to RSV, lower respiratory tract infections, in South Africa and in other low and middle income countries. The disease that we decide to call asthma is different disease in different parts of the globe. But importantly, lung function gets really impaired with any wheezing episodes. Don't forget that. So what you can see is quite interesting story. Machine learning, much better performance over regression-based model to predict asthma, but really we're not there in terms of being able to roll it out into clinics. There clearly are distinct both temporal and pathophysiological clusters of preschool wheezing, and if you come to the course, you will hear a lot more about this from Sejal, myself, and Andy. They are underpinned by unique genetic architecture, but are not associated with diagnostic labels which we use in clinical practice. And that's why our diagnostic labels are not good enough. Importantly, all of these clusters are associated with impaired lung function, even a transient one. So what we need to do is move forward towards understanding determinants and mechanisms of these different subtypes of the syndrome we agreed to call asthma, and ultimately reform the taxonomy of childhood asthma from symptom-based towards mechanism-based. And you can do it. You can do it from vast amounts of data on clinical observation, scrutinizing by improving analytical models, target prediction modeling, regression modeling, but also unsupervised modeling to understand patterns in the data and then follow this with carefully designed experiments. It cannot be done by a single person. This is just several people who have contributed vastly to this. Colleagues from Unicorn Birth Cohort Consortium, Angela Simpson-Clare Murray in Manchester, Raquel Granell in Bristol, Steve Turner in Aberdeen, Sejal and Paul here, John Graham and Hassan in Southampton. Our extraordinary data science team led by Dr. Sarah Fontanella, absolutely fantastic one. Our collaborators in Microsoft Research across various birth cohorts, Claire Lloyd, Seth Johnson, who do all the mechanistic experiments, and all friends and colleagues, particularly Heather Zar from around the world who are contributing to this. And with that, I thank you all very much for actually joining us for this today, and I'll be happy to answer any questions that you may have.
Thank you so much, Adnan. You packed so much into that lecture. It's mind-blowing the amount of work in there. We've got really little time for questions. As I said, there'll be more in the June Q&A session. One quick question, I don't know if you can answer it quickly, Adam, but from one of the people online, what are the latest developments in controlling and treating asthma? So in controlling and treating asthma, it's quite a tough one to answer quickly, but
I mean, our real problem is our guidelines. You know, when you think about medicine, you know, there are two types of guidelines. One is stratified, which is mechanism-based, which is personalized, and another one is one size fits all. And our guidelines for asthma, despite the pretense that they're not, are still really sort of predominantly one size fits all. Try treatment, and if it fails, try another one, and then try another one, and then try another one. There will be a new report from European Respiratory Society Task Force on Preschool Wheezing coming, I think, in 2 to 3 months, giving you clarity about how we can stratify patients. And if you join us, of course, Sejal is going to be talking about this.
Lovely. Thank you. That's a great answer. So thank you again, Adnan, for an amazing tour de force. We're going to move on to Marta Vazquez-Ortiz, who heads, I think, the largest consortium around FPIES, a particular form of food allergy in the world. And so she's in a great position to tell us all the latest information about food and protein-induced enterocolitis syndrome and what it means for people. Thank you, Marta.
Thanks so much, Bob, for the kind introduction, and thanks everyone for being there. So I think we're a bit short of time and I have quite a few slides, so I'm going to try my best there. And it's always difficult to speak after Adnan because he's such a great speaker. So this is what I'm going to be talking about today. So the FPIES Research Network, which is the collaboration we set up in 2017, and the work we've been doing within that network. So first, gathering the FPIES community's views on the challenges and unmet needs for patients and families living with FPIES, and then our work trying to improve FPIES diagnosis in a number of areas, and then work around infant feeding. How can we support families in that tricky time? And then some very exciting and very recent basic science results, particularly in the area of genetic predisposition to FPIES and pathways, mechanistic pathways. So starting with the network. So the network is a collaboration between Imperial and GenVip in Spain, which is a very strong team in pediatric infectious diseases and a long-standing collaborators of the pediatric infectious diseases team at Imperial, particularly looking at omics for diagnostics in febrile disease in children. And it was back in 2017 through an idea from Bob, actually, that we set up this collaboration. And we have 14 recruiting centers in Spain and Italy, basically recruiting children mainly, but also adults undergoing FPIES challenges, particularly over follow-up. And, lots of people to acknowledge there, but key people, Alberto, who leads the basic science side of things in Spain, and then our PhD student and the Imperial Data Science team led by Sarah, as Adnan mentioned. We have a number of funding sources, and more recently the FPIES research grant from last year, which is really exciting. So the aims when we set up this research collaboration was primarily to investigate the biological mechanisms underlying acute FPIES with the aim to identify diagnostic and prognostic biomarkers using a multi-omics approach, again, using the expertise of the colleagues in infectious diseases, and Bob had done some pilot work around that on RNA-seq. But also, when you gather this really valuable cohort of patients, trying to assess the clinical characteristics of this South European patient cohort and what we could learn to help improve current diagnosis and management of acute FPIES. But also, and more importantly, probably, to understand the patients and the families' challenges and unmet needs, to try and co-design strategies to improve the clinical care and to improve research strategies for FPIES. So in terms of the study design of bio-FPIES, it's an observational study. And as I said, we recruit patients with a clinical diagnosis of acute FPIES undergoing routine oral food challenge in clinical practice, generally for follow-up. And we basically collect clinical data and quite a few samples on the challenge day to everybody at baseline, and then if patients react at reaction onset and 4 hours post-reaction. And we collect blood samples and saliva samples, so quite a few tubes, as you see there. And we've done quite well in the last, 6 years, taking into account there was a pandemic, etc. And we've managed to recruit over 300 patients undergoing challenges, 1/3 of them reacting at challenge, so over 100 reactors, and then over 200 non-reactors there, and a big sort of biobank with lots of tubes to analyze, which is really exciting. And then moving on, one of the first things we did was to collaborate with patients' organizations. So FPIES UK, the FPIES Foundation has been very generous and very collaborative from the very start, and the team in Australia to try and run a survey on parents, on patients with FPIES, trying to understand what are the top research priorities or top priorities for them tackling their needs. And it was interesting to see that it was very much around improving diagnosis and prediction of tolerance. And you see that in order of importance, better awareness amongst healthcare professionals and medical students, and then tests to predict resolution, safe foods, and tests to diagnose FPIES acutely. So a long way to go still on pretty basic stuff for the disease. We did qualitative research and with thematic analysis, looking at the challenges and the needs. And some of that is quite similar to IgE-mediated food allergy, but probably, it transpired that people feel much less understood. And there are many more unknowns in FPIES, which I think nobody will be surprised about, but lots about anxiety and stress. So we got lots of really lovely quotes there, social restrictions and discrimination, lack of knowledge and support. And then in terms of needs awareness, information, education, and research to improve knowledge. Okay, so lots of work to do. So moving on to the work we've done trying to improve FPIES diagnosis, I'm going to start talking about diagnostic criteria and things. Okay, so clinicians and also families as well dealing with FPIES, we know the diagnosis is not easy for those who are not very specialists. And one of the first things is that there's limited awareness outside allergy specialist centers. And the reality is that FPIES does not really look like an allergy or the conventional sort of immediate allergy you're expecting. It's predominantly gut symptoms, plus minus systemic compromise, it's delayed, it's relatively rare, it's some kind of suspicious foods like milk, fish, or egg, but also unusual foods like fish like veg or fruit or rice. and it has negative allergy tests, and the allergy drugs are ineffective. You don't have any diagnostic tests, and you basically rely on a clinical diagnosis of recognizing the clinical picture, the typical one with a projectile repetitive vomiting, often with pallor, lethargy, etc. But the differential diagnosis is broad and complex, and again, awareness is not great. So that leads to delaying diagnosis or misdiagnosis, many reactions for patients. So lots of impairment in quality of life, impact on safety, etc. So how do we diagnose FPI? So, many of you will be aware of the consensus in the international consensus on FPI diagnosis and management that was published in 2017. So these are, for now, the best diagnostic criteria that you can find. So you have to have FPIs, you have to fill the major criteria, which is vomiting, usually within 1 to 4 hours after eating the food, and then fulfill 3 or more of the minor criteria, which is, vomiting more than, in one occasion to that food, or reacting to a different food with FPIs, or having a lot of lethargy, pallor, etc. So quite a few things there. Okay, so when we started via FPIs, the consensus had just been published, and we said we're going to follow criteria. The patients that come in, they're going to have FPIs, we're going to follow criteria. And what we realized with the first 50 patients we recruited was basically that 1 in 4 didn't meet criteria, and they all had repetitive vomiting episodes in multiple occasions to the same food, but they wouldn't reach the 3 minor criteria. And it was very much those with a milder phenotype that just wouldn't tick criteria. Okay, and we compare with other criteria in the literature, and we kind of flagged some limitations there. At the same time, some colleagues also from Spain had published some series of FPIs in adults, which is, a more recently sort of described phenomenon. And quite interestingly, 40 to 60% of adults with FPIs don't vomit. They might have lots of nausea and abdominal pain, which is the key phenomenon, but they might not vomit. And that led us to propose some revised criteria, still with the major criterion of vomiting, which in adults might not be present, and just nausea might be present. And then in terms of minor criteria, we thought that reproducibility is a key feature that might give you more points than other things. Okay, and abdominal pain, probably in older people and adults, is probably quite present. And we also saw in FPIs, as I will show later, that neutrophil increase in reactions is a very, very common phenomenon. So we introduced that there as well. So some work on that. So another interest we've had over the years is trying to understand if IgE testing is helpful. And there's some really nice papers from the last 4 or 5 years on the FPIs pathophysiology, lots about data-driven analysis, etc. And IgE doesn't seem to be involved in the pathophysiology. So the question, very clinical question, is do we have to test or not? And the consensus gives you slightly sort of mixed advice, suggesting perhaps in milk FPIs might mean a more persistent cause, or there might be a potential for switching to IgE-mediated allergy. So we looked at the literature, and we saw that in the last few years, there have been quite a few papers touching on that topic of IgE in FPIs. So we decided to do a systematic review about the role of sensitization in acute FPIs. And this has been the MSc thesis of one of our students, who's an allergy dietitian, to submit it today. So very exciting. So this is probably the first time I'm sharing this data, and we will submit for publication soon. So basically, there are 53 studies that survived the screening, so assessing skin prick test or IgE to the culprit food in acute FPIs. And basically, the questions we had were, okay, do I need to test at onset when I diagnose patients? Do I need to test at follow-up? What does it mean if I have a positive? Does that mean that they're going to have an immediate reaction? Does that mean they're going to have a more persistent cause? So these were the questions we wanted to answer, if we could, with the systematic review. Okay, so what we find out, basically, sensitization to the culprit food in acute FPIs happens in 11% of patients, and it's higher in egg and nuts. And there are some reports suggesting that there might have been an increase in egg and nut FPIs in the last few years in the context of early nut and egg introduction for IgE-mediated food allergy prevention. So that's quite interesting. Seroconversion, so turning from, say, negative to positive allergy tests over follow-up, only happens in about 2% of people with FPIs. What we call a phenotype switch, so moving from having acute FPIs reaction to IgE-mediated reactions, only happens in about 1% of people diagnosed with acute FPIs. And if you just look at those who are sensitized, only in 8% of them. So that means that about 90% of people, most studies were in children, who have IgE to the culprit food, they will continue not to react with an immediate reaction. Okay, and in terms of association of disease persistence, we found 12 studies with an analytical design, and 4 of them had found an association, but 8 studies had not found any association. And there was no clear difference in terms of, one group of studies being stronger than the other. So for now, I think we can say probably as before, doing the systematic review, but with much stronger data, that, you're expecting a negative result, but if it is positive, very, very unclear what that adds in practice. And I don't think we have any sort of strong evidence to really advocate for doing testing or acting on the test, really. So moving on to challenges. So and looking at, how can we improve the safety and the diagnostic accuracy of challenges in FPIES. So if you look at, again, the consensus, how to do challenges in FPIES, very sensible advice and recommendations there in a medically supervised setting with rapid access to fluid resuscitation, prolonged observation, because we know, serious reactions can happen. When you look at the specifics, of the protocols to follow, and particularly with regards to the doses, there's this advice of using this 0.3 gram protein per kilogram in 3 equal doses over 30 minutes with a maximum of 3 grams of protein. And when you try and do that in practice, it's quite problematic. Mainly, the 3 gram issue, if you have high protein foods like fish and egg, which are the leading culprits, say, in Southern Europe, you're not reaching by far an age-appropriate portion, okay? And if you're dealing with veggies or fruit, that's going to be a huge amount that you shouldn't give, really, okay? And I guess we're also concerned about just giving the full dose within 30 minutes, because, if this is a disease that takes kind of potentially hours to develop, you've given the whole lot. So again, we decided to try and learn from what was published, and we did a systematic review on challenge procedures in FPIES, again, with one of our MSc students, Taisa, who's based in Qatar and is an adult allergy specialist. And the aim was to describe the different methodologies and clinical outcomes, but particularly to try and identify features of the challenges that might be associated with better safety and diagnostic performance. So we gathered 51 papers after screening. Again, strong methodology. We had Ulukbek, Marta Vazquez with the other systematic review helping. And we saw there was great heterogeneity in the challenge methodologies, the clinical outcome reporting, not much in terms of data about certain clinical outcomes like severity or treatments used. And we were not able to conduct meta-analysis, but we did a narrative synthesis. And I'm trying to present the key findings that might be helpful when you're trying to decide how you challenge your patients. Okay, so we saw that the few studies that had given a relatively not too big dose to start with, were reporting that 25 to 30% of an age-appropriate portion triggers a reaction in most patients, and that's over 70% of patients. So you shouldn't be giving up an age-appropriate portion, say, to start with. Okay, and then that over 50% of patients react at least after 2 hours. So you need to, give them time. But the key question we wanted to answer is, can we tell if dose matters for severity in FPIES? We couldn't find any RCT, no prospective head-to-head comparison of different protocols, and there was a lot of heterogeneity in outcome reporting, such as people using different classifications of severity, so very difficult to compare. There was the paper by Infante, who's a close friend and collaborator as well, so from Madrid, on fish FPIES, and they basically compared a historical challenge protocol where they used a 1-day approach with multiple increasing doses, as most centers will do, with a cumulative dose of an age-appropriate portion, and they compared that to a new protocol they implemented subsequently, where in day 1, they only gave 25% of an age-appropriate portion as a single dose, and then on the second day, they gave an age-appropriate portion if the patient had tolerated the previous one. And basically, they found that that new protocol had a lower rate of severe reactions with statistical significance. So we look at the BioFPIES data, and we have now incorporated that to the systematic review and have sent for publication recently. And this is what I'm going to be talking about now about BioFPIES. So we looked at the first 100 symptomatic children at challenge, of whom 81 met criteria for a positive challenge, and 23 of them were severe. 88 had undergone a 1-day protocol with multiple doses in 1 day, and only 12% had gone a 2-day protocol, which is exactly like Sansone's protocol for fish, with 25% on the first day. And then when we looked at the subgroup comparison, those with mild, moderate to severe reactions at challenge, the main difference was that the 2-day protocol group had less severe reactions, and there were no differences in baseline characteristics and no other differences in challenge features. And then we did logistic regression, and again, that was an NEC project for a student, Maria, who had a background in stats and maths, so that was very helpful. And we ran an analysis there with, you see, a number of variables there, and the only independent factor influencing severity at challenge, so predicting whether you're going to end up having a severe reaction at challenge, was doing the challenge in a 1-day protocol as a risk factor, as opposed to a 2-day protocol, which was protective. So dose and timing seems to matter for severity, but we need prospective sort of randomized controlled trials to sort of prove that. And then another thing we wanted to look at, because there are no data out there really, is like, how does severity develop over time? In IgE-mediated food allergy, we're all very worried because we cannot predict the severity of future reactions, and that impacts a lot on clinical practice. So what about FPICE? So we saw that 70% of our patients, these 100 patients, qualified as severe reactions at onset, okay, mainly because of very significant lethargy as young kids. And then at challenge, most of them, over 50%, had less severe reactions, and only 12% had more severe reactions than before. So when we look at the kids who ended up reacting, only 1 in 10 ended up having a severe reaction without having had a severe reaction before. So I think in a way that's reassuring, but of course it's just, a bit of sort of just very observational data. So the next thing I want to talk about challenges is how we assess the outcome. And again, the consensus has criteria for that. So vomiting, and then you need to meet 2 or more minor criteria there. And when we apply that to the 100 patients in this bio-FPICE with symptomatic challenges, we saw that in terms of the positive ones, pretty much all of them had increased neutrophils, so that seems to be pretty much universal. Abdominal pain, which is not within the minor criteria, was common, was 43%, so might be useful to include, particularly, as kids get older in follow-up challenges and might be able to report it. But diarrhea, which features within, the criteria, is not there. But interestingly, out of the 100 symptomatic challenges, 19 didn't meet criteria, so they were left as inconclusive. And when we looked at them, those 19, they all had objective symptoms suggesting FPICE. 15 had vomiting, 4 didn't vomit, and they were all the children. So we wonder if that was sort of a bit more like, as adults do, not vomiting. Neutrophil increase in all of them where we tested, and it was mostly mild reactions. So they kind of don't manage to tick the box, and they had milder reactions based on background histories. It seems to be perhaps a milder phenotype, that they're not going to react in a, more severe way. So you meet you tick the boxes there. Okay, so, just applying a bit of, clinical judgment, likely positive, and, being aware of the criteria, but also potential limitations there. And what we saw in these 81 positive challenges is that, neutrophil increase is pretty much universal, but the changes in the full blood count are pretty much, affecting also eosinophils, lymphocytes, basophils, which go down, particularly at the 4-hour post-reaction start time points, and the hypothesis for that potentially moving to the periphery there. And the last thing about challenges, we with the negative challenges, so those who didn't react, we call them at 48 hours and a month later to double check they're tolerating the food. And we did that 3 years ago with the first sort of bunch. And we realized that in kids with negative challenges, 1 in 8, they had reacted with an acute FPICE reaction the very next time they had tried the food at home, okay? So we understood that as a false negative of the challenge. There's been then communications about, is this, recurrence of FPICE? We really think, we might be missing some patients with that just kind of single sort of exposure. So we kind of need to be aware of that and potentially warn our patients when we discharge them from negative challenges. So that's, much quicker on infant feeding, a bit of work there. So we're very aware, in clinic, often when you see infants who have had quite bad reactions, the parents, of course, are very worried about trying new foods, having similar experiences, etc., which is, of course, very understandable. So we wanted to understand a bit more what's this kind of their experience there. And last year, again, with another MSc student, Imelda, who's a pediatric trainee, lovely girl, there we did an international survey, again, collaboration with the patients' organizations on over 100 parents. And we asked a number of questions, and I was very interested in that question. So how frightening was the initial reaction and weaning your child with F5s? And most parents, you could say not frightening at all, okay, but most parents said extremely very frightening, which is, quite telling. About a third reported significant feeding difficulties, and only about half had advice for that. And those with feeding difficulties reported a higher impact in quality of life and well-being. And independent risk factors for having or reporting feeding difficulties was a number of foods involved, faltering growth, which are well-known risk factors for feeding difficulties, and also being frightened or very frightened to feed your child. So that kind of interaction that develops when you're very worried seems to influence sort of the feeding difficulties and how things develop, which is also known in other fields. So definitely an area for support. And we asked about what would be helpful, and parents suggested some useful things like online fact sheets, guides, videos, etc. So a question we've had, and again, coming from Spain, you train seeing that, we don't give specific advice about solid food introduction to kids with F5s because it doesn't seem like they react to all things, okay? So when you hear the term multiple food F5s, or at least when I was in training, it was like, okay, I don't see that often, okay? When you look at sort of the English literature, it's quite common, particularly sort of more in tertiary centres, etc. And there's some good papers like correlating different foods. And this study from Australia was a population-based study, and even for milk, they reported a 44% rate of multiple food F5s. So quite interesting. And based on that, there are kind of some guides around, how to try foods like high risk and lower risk, etc., which I think can be helpful in this context. When we looked in bio-F5s, we went kind of, we asked the kids, every kid one by one, all the different food groups and whether they tolerated. We look at that in a lot of detail, looking for F5s, but also other forms of food allergy. We saw that multiple food F5s was happening only in 5%, and it was the kids reacting to, say, the very uncommon foods like veg, fruit, etc. So similar to what had been reported in Australia. And other forms of food allergy, which is also pretty rare. Okay, so probably in that sort of setting, and there have been more recent similar publications from other countries, sort of similar sort of Mediterranean region. So you might not need complex feeding advice in that sort of area. And that kind of fits into, we know with F5s there are quite a lot of geographical differences, so that could be another one, okay? And this is the very last page, a couple of slides only, some kind of exciting data from that sort of biobank and the colleagues in Spain and the data science team at Imperial. So this is work from the GEMB group in Spain with one of the grants we got some years ago. So on whole exome sequencing, so trying to understand the genetics, to try and understand disease susceptibility and pathophysiology. And we did in 41 patients with challenge-proven F5s from bio-F5s, whole exome sequencing association study. So we've just sent it for publication recently, and really exciting results. And I think, as a clinician, you kind of, the clinical data, you kind of see it coming, but, this thing, you kind of see it all at once. So it's really, really interesting. So 3 genetic variants, so SNPs associated with F5s, one in a gene that is associated with this uncommon syndrome, thrombocytopenia absent radius syndrome. And I, as an allergist, had never seen it, but when you look at the literature, up to 2/3 of cases associate cow's milk allergy with vomiting and diarrhea. So these are classical series, but when you read it, you really wonder about F5. So really interesting. And my colleagues have done some additional analysis, and they seem to see a functional correlation between this gene and filaggrin, which is within the same domain. And then 2 variants in a gene that is strongly associated with inflammatory bowel disease. And then 6 other genes associated with F5s with further analyses, and 2 particular genes looking very interesting. One involved in induction of TGF-beta signaling, which is crucial for epithelial barrier integrity and IgE production, and has TGF-beta has been already associated with F5s. And then another gene which is involved in immunoinhibition receptors in immune cells. So quite interesting, potentially helping us understand the genetic basis of F5s, and the fact that probably the pathways contributing to F5s involve epithelial barrier dysfunction and immune dysregulation, as it's been seen for other allergies and IBD as well. So quite interesting. And I'm just going to go very quickly through this. So some preliminary data on transcriptomics, and I would like people not to share because we haven't analyzed this in depth, and we definitely haven't published. So this is RNA-seq data on 50 positives and 25 negative challenge people from bio-F5s, and both longitudinal analysis in the positives, but also comparative analysis at baseline between the positives and the negatives, showing some differentially expressed genes involving immunological and sensorial pathways there. So quite interesting and more work to do. And then some nanostring data, microRNA in blood and saliva, also showing some really nice data there in terms of potential immune mechanisms involved, including in saliva as a proxy and more sort of less invasive sample. So quite interesting there. So this is the summary. So we've touched on different areas within BioFPIs and hopefully more to come. And we're very, very grateful to our funders and particularly FAIR for the very generous grant that is going to allow us to look in quite a lot of detail about transcriptomics and proteomics in the next couple of years. So thanks so much and happy to take any questions.
Thanks, Marta. So I think hopefully people can appreciate the strength and depth in allergy research at Imperial. Again, you packed in so much into that short period of time. So, and thank you for trying so hard to catch up on time. Again, time probably just for one question. Someone's posted, what dose do you use for fish FPIs? So when you're doing a food challenge for FPIs, how much fish do you give people to prove that they've grown out of it or not?
Yeah, so we've in the clinical service, I mean, Bob probably is laughing because we spent a bit of time in the last couple of months like revising our challenge protocol, and we basically decide based on the clinical history, and we're quite sort of risk averse in the team, I think, and we start quite low, particularly in the children who've had like life-threatening reactions. For instance, we start pretty low. We start, I can't tell from the top of my head because we just have redone the whole thing, but I believe we're doing based on grams of protein, starting quite low for that. So I think there's so much heterogeneity, and at the end of the day, I think the clinician needs to be comfortable with what they're doing. And I think within our team, there's a lot of people. So we had people saying, "But I trained, I really trust the system," and it was 50%, of the age-appropriate portion, and then a couple of hours later, the other 50%, and I'm comfortable with that. And other people wanting to start with 3 milligrams of protein, which is nothing to do with that. So I don't think I have a straightforward answer. But yeah,
that's a great answer in itself. So there isn't a consensus on that, really. Thank you so much. I think we'll probably leave it there because we've gone well over the hour. But just a reminder to everyone. So just come back on the scholarship question. Those scholarship numbers I put in the slide, they are confirmed now. I'm sorry, Marta, let me know that. And see you on the 5th of June. Anyone who wants to come and ask more specific questions about the master's program. Thanks everybody for your attention.